Mm. Khazima, I think we should start.
ठीक है डॉक्टर समर डॉक्टर नाजला should we start? Do I have your permission? Okay, I guess that's a yes.
Yes, I forgot to unmute my microphone.
No problem, no problem. Good day and assalamu alaikum, ladies and gentlemen. Thank you very much for joining us. We are formally starting today with the second day of our online certificate course on basic plant tissue culture techniques. And we will start today with a lecture from Dr. Najla Abdullah. The title of her lecture is Basic Tissue Culture Techniques and Their Application. Let me introduce Dr. Najla to you. Dr. Najla is a professor and Department of Genetics at Faculty of Agriculture, Cairo University. She is director of Egypt Biotechnology Information Center and part of a growing network of biotechnology information centers of ISA's Global Knowledge. In 2009, she was appointed to be the editor-in-chief of GM Crops, the first international peer-reviewed journal of its kind, launched to focus exclusively on genetically modified crops. Dr. Abdullah is a member of Institutional Biosafety Committee at Faculty of Agriculture, Cairo. Her research focuses on plant molecular genetics and bioinformatics. She is particularly interested in developing transgenic plants resistant to pathogens and better suited for the environmental challenges. And with this, I request Dr. Najla to please start her lecture.
Good morning, everybody. Assalamu alaikum. It's my honor to join this wonderful workshop, and thank you for the organizer for inviting me for such workshop. Today, I'm going to talk about try to make a basic technique for tissue culture and its application. So I'm not sure if you can see my my slides or not. Is it clear?
See your slides.
Yeah, okay. So thank you for the introduction for me. So I'm going to talk about a basic topic about introduction, history, basic technique for tissue culture, its application, benefit, and constraints. So I think you already started yesterday, so you already know what the meaning of tissue culture. We're just going to mention that tissue culture is an in vitro cultivation from plant cell or tissue or organ under aseptic condition, control condition, into liquid or semi-liquid with media containing different nutrition, just to produce regenerated plants. So it's tissue culture, which does not move, does not switch. Okay. So, of course, in vitro means that we have to cultivate the plant in tubes or jar or petri dish or plastic bags and so on. And under aseptic condition, that means that we have to use a micro-free condition. I know what's wrong. Okay. It should be 100% micro-free. It should be autoclaved in an oven or ethanol treated or Clorox treated. So why do we need to do tissue culture? If we want to have exact copies of the plant we have to use tissue culture. Sometimes we don't have enough plant to propagate in the field, so we need to propagate it in vitro. In vitro tissue culture, that will be very quick compared to cultivation in the fields. We can have from one plant multiple copy of plants, so we can do regeneration from one plant to from one cells to whole plant. We can also use it for production without going through the seeds. Okay. For the historical part, I don't want to go through it because it's a little bit boring. So the first trial was in 1902. They tried to cultivate using different culture from meristemic cells, but they failed. In 1939, they were able to develop callus by Gatheriet from carrot. Schock and Miller were the first to, to produce forced hormone hypothesis. In 1966, they obtained haploid plant from pollen. In 1991, they get flower from bamboo. In 1966, they have haploid plant from anther culture. So, okay. So what is the basic requirement for plant tissue culture? Of course, we need plant material. We need equipment like laminar flow incubators, autoclave, sterilization for sterilization, and refrigerator. We need aseptic condition, washing and the storage room. We need media preparation room, sterilization room, nutrition media, transfer room, culture room, or incubator, proper or optimum aeration, and so on. So the major factors affecting the process of regeneration, first of all, we have a good source of exoplants. So this exoplant should be very active in dividing cells. So it should be from meristemic cells. So you can use from any part of the plant, from organ or different organ, to get the suitable exoplants. Of course, exoplant can be different from one species to another. So we have to choose the right one for each species. We need cultivating culture environment, like pH, suitable pH, suitable light, humidity, temperature, and so on. We need nutrition, media, and constituents. So, as we said, we need meristemic cells to use it for tissue culture because it's going to be divided in synthetic media. Already developed or aged cells cannot be used because they're very slow to divide. The second important thing is we should have disease-free plants. So first of all, you have to have a good sterilized exoplant to start with it. So sterilization is very important for starting the tissue culture. So what's the plant material? We can use organ culture, any part of the plant, root, tapes, shoot tapes, leaves, so on, flower, any type of organ, or we can use hairy root or protoplast culture. So if we use organ, if we use shoot formation, we need to develop root. If we use root, we need to develop shoots, and by the end, we will have the whole plant from regenerated plant. So there is two types of organogenesis. We have direct organogenesis and indirect organogenesis. So direct organogenesis, we can use an organ and developed the other part of the plant. And this, if we use auxine, that will allow to induce root. If we use cytokinin more than auxine, that will form shoots. So this will be direct regeneration. Indirect will go through tissue culture. Sorry, it will go through callus. So it depends on the auxine and the cytokinin. We can have shoots or roots, as we see here in the picture. If we have many cytokinins, we will have shoots. If we have auxine, we'll have roots. If we have balance between shoot and root, we will have callus. So direct organogenesis, we can have exoplant, meristemoid, perimeroid, and the indirect will have callus in between. So what is the difference between direct and indirect? For regeneration, it doesn't matter, but for developing modified plant, it's better to have direct organogenesis to avoid chimeric plant. The other type is embryogenesis. Embryogenesis, that means that we will have non-zygote embryo. So we can have any type of cells or group of cells that can form embryo, but not from the zygotes. And also in the embryogenesis, we can have direct embryogenesis and indirect embryogenesis. Indirect, of course, it will go through the callus. The media composition, I think you already have it in the practical part, but the main component of it is macromolecules, carbon source, vitamins, growth regulators, which is very important for the cell to divide, and gelling agent to have it semi-solid media. So the basic technique is to prepare a suitable nutrition media, autoclaved of course, so we don't have any contamination. Select the type of exoplant according to the species we're going to use, and even sometimes within the strain itself, it can have different genotype better than the other. So we can select this, the exoplant. It has to be sterilized, so we will not have any contamination. Incubated under aseptic condition with suitable nutrition. We have then we will develop regeneration. We have plantlet, shoot with and root, either going through callus or without callus, direct or indirect. And then after we have the plantlet in the jar, then we need to acclimatize them. So we need to harden them by a different environmental condition before we transfer it into the greenhouse. So this is the main steps. We have the exoplant cultivated, have the shoots and the roots and so on until we have the plant, the full plant already available. This is a very short brief about tissue culture. Now we're going to talk about application of tissue culture. Tissue culture, we can use it for micropropagation, for germplasm preservation, for forming synthetic seed, for micrografting, for disease and unhealthy plant elimination, for secondary metabolic production, and for genetic improvement. So all of them is direct application. This is an indirect because you use a tissue culture as a tool for performing modified plants. For my micropropagation, of course, as we said, we can have some part of the plant and we can develop a huge number of the same plants. If we have different difficulties for cultivating this plant, or we have a very small amount of seeds, we need a mass production of it, so we use micropropagation. For virus-free, if we have a plant, a very precious plant, we need to preserve them. So they should be virus-free, and we have to use different technique to avoid the virus in this plant. For the secondary metabolites, this is usually used in industrial purpose to have mass production of the desired products. So for micropropagation, we can use shoot, roots, or any part of the plant, and then grow them in under growth regulatory materials. As we said, we can use cytokine or auxin if we need to form shoot or root, then transfer them into suitable media, and then acclimatize them and use them for different purpose. We should avoid using an exceed number of plant growth regulation, because this will cause us a problem. Like somaclonal variation, and this is undesired feature in the tissue culture, unless we do it in purpose to have mutation. So this is the different steps. We have the mother plant, we have to have a shoot from it and root, and to have mass production of this plant. We can cultivate it later in the open field or in the greenhouse. So the second application is to have a virus-free plant. We can use the shoot tips because it has a lot of meristemic cell, very active. And if we cultivate this shoot tips, we depend on the cells can divide more than the virus replication. So we can choose the active shoot tips for cultivation and to eliminate the virus from the developed plant. This is a very useful technique, and already have been applied in different varieties of plant species for different kinds of viruses, and it's very well established technique. The second part is somatic embryogenesis, synthetic seeds. So if we want to preserve different plant or use it for later, we can make like a synthetic seed. So we can have the small plantlet, juice, or any part of the plant that's very active and coated with suitable materials. We can keep it on the shelf or in cryopreservation for a long time. And when we want to cultivate it, we can use it for cultivation, and we have the plant ready with mass production. The other application is micrografting. If you want to have the root from one plant and the shoot from other plant, we can use micrografting by grafting the shoots from one part of exoplant with the root from other part from exoplant in the tissue culture level, and we will have the grafted plant ready for cultivation. The other thing for applying for tissue culture is the secondary metabolite production. So this technique is very important for the industrial purpose. They have been used for several years for production of different industrial and medicine products, like a quinine for malaria. So we can use the plant for can use isolate the quinine for use it for curing malaria. The cyclonine is antibacterial agent. We also can isolate it from the plant. The piperine is used as a drug for diabetes, for high cholesterol and high blood pressure. All these techniques can be used in as a secondary metabolite. Besides, we can also allow the plant to develop different drugs we need by producing a modified plant with the gene desired that can produce these desired products, like vaccine and other kind of drugs. So the different steps we use plants, we can take the exoplant from it and we use it for either producing for direct or indirect through callus. And then we have cell suspension. We can use large fermenters to have a mass production. Then use an different technique for isolation, like HPLC or different kind, and we'll have the drug ready for marketing. The most important part, which I like most, is the genetic improvement plant. So genetic improvement plants, we can use different technique, like genetic transformation or genome editing. Genome editing and genetic transformation depend on modifying the DNA to have a proper plant that can be that can carry useful characteristics that we need to share or to use for our life. So the first part is the genetic transformation, and this can be performed most commonly by agrobacterium transformation or biolistic transformation. Both techniques need tissue culture. So tissue culture is a very important steps. Without them, we cannot have genetic transformation, or sometimes we don't have genome editing, although some genome editing technique can avoid using tissue culture. So for developing transgenic plants, we need to deliver the DNA into the plant cells. Then we need to integrate the DNA into the genome. So once it's entered the cell, that doesn't mean that it is transgenic. It has to enter inside the genome and be stable, transform it stable. Because if it is entered inside the cell, we call it transient expression. It will not be inherited through the cell division. And then from this transgenic plant, we need to regenerate them and select the transformed cells from non-transformed cells and produce the whole plant tissue from this kind of cells. So the first technique is using agrobacterium transformation. Agrobacterium is a natural soil-borne bacteria. It's a pathogenic, unfortunately, but it uses natural transformation. It introduced part of its plasmid inside the genome of the plant cells. So it's not only introduce the DNA into the cell, but it also integrate the DNA into the nucleus and in the chromosomes. So there is different kind of agrobacterium, but the most commonly used is Agrobacterium tumefaciens. Sometimes we use rhizogenes, which form hairy root. Agrobacterium tumefaciens form crown gall, which is a pathogenic character, where we isolate the pathogenic gene and replace them with a desired gene. So if we want to develop transgenic plants, we use the exoplants. We prepare the exoplant, we prepare the agrobacterium with a desired character and incubate them together, then cultivate them into selection media, which allow the transgenic plant also only to grow, and non-transgenic plant will be eliminated. Then we have the callus, if we have indirect, and we have the shoot and the root, and this in the end, we have the putative transgenic plants. The other part, the agrobacterium transmission is usually work well with dicot plants, but monocot is sometimes very difficult to use agrobacterium. That's why they develop another technique to deliver the DNA inside the plant tissue, like the Biolistic bombardment-mediated transformation. This is a gunning DNA. We coat the microcarrier with the DNA and stick them with sticky material as spermidine, and use the bombardment to allow the DNA to enter inside the cells. So through the gun cells, when it's fired, the microcarrier which carry the DNA will enter randomly into the cells. Some of them will remain in the cytoplasm, some of them will enter the nucleus, but very small amount will enter into the chromosomes. So the efficiency of transformation with the Biolistic gun is not very efficient like agrobacterium, because we lose a lot of DNA into the cytoplasm and into the nucleus without integrating into the chromosome. That will allow forming chimeric cell. Chimeric means that some of the cell is transgenic and some of them does not have the desired gene. Okay. Now this is the main technique that we use for transformation. So how do you know how where biotechnology crops are cultivated? Most of the country cultivate transgenic plant is the United States and Canada, and we have 10 countries in South Africa or Latin America. Latin America. We have about 8 countries in Asia. We have 6 countries in Africa and only 2 countries in Europe, because Europe is anti-GM, of course. So GM, although we have a huge amount of plants that transgenic that has useful characteristic, even in our country in Egypt, we have a lot of plants ready in the shelf, but we cannot use it due to the biosafety regulation. So biosafety regulation is the main concern that does not allow us to use or to apply genetic modified plants. So we have here about 41 countries already practice using genetic modified. Not all of them are cultivating right now, because some countries does not no longer cultivate GM crops, like Egypt, like Burkina Faso, like Iran, and so on. This all countries already cultivated in the previous year, but they no longer use it because of the biosafety regulation restriction that does not allow them to use. So here we can see the different crops that cultivated in each country. We can see that there is a huge number, about 22 crops are already used for commercialization and cultivation in the open field. So in 2019, I'm sorry, this is an old data because I don't have yet the new one. We have in North America, they have different crops like maize, soybean, and so on, canola, sugarcane, papaya, and so on. Latin America, they have also different crops. In Asia and Europe, those are the example of different crops are cultivated. In Africa, we have only maize, soybean, and cotton. In Europe, they only have maize. So if we see the amount of transgenic plant cultivated for each crop, we can say that cotton, 79% of cultivated cotton worldwide are transgenic, only 32 is non-transgenic. Soybean is followed because it has 74% of them is transgenic, and followed by maize, which have 31%. Canola is 27% of the worldwide are cultivated with genetic modified. Now we have some example of biotech crops, which are very useful, and some of them are used for food and feed, not like the other one for disease resistance or herbicide resistance or biotech stress resistance. Recently, they already use it for food and feed. Like in potato and apple, they have transgenic plant for browning, against browning. So they can stay in the shell for a long time. And if we cut this plant, they will not form brown culture. Also, one of the main very good example is Bangladesh, which cultivate eggplant for disease resistance, and they are used for human consumption. Also, we have alfalfa lignin, low lignin, also is cultivated in US and Canada and Argentina. And for sugarcane, we have plant resistance for insect and for drought tolerance. For sunflower, we have high oleic acid. For the pineapple, we have pink pineapple in Costa Rica. This is very nice plant. And also we have lettuce, non-browning lettuce. We have Colorado beetle-resistant potato. We have drought tolerance wheat. We have drought tolerance, salt tolerance rice. These are some examples of transgenic plant. Of course, there is a huge number, but those are very important and used for food and feed. Okay, the main 5 countries that cultivate or plant biotech crops are United States, Brazil, Argentina, Canada, and India. We can see that the cultivated area is about 190.4 hectares in 1990, which has a reduction of or decrease about 1.3 million hectare. This is because of some plants does not are not acceptable by like soybean. The different country that does not allow exporting this kind of plants. And because of the rise of another modified plant like the genome editing, a lot of countries preferred using genome editing rather than using genetically modified or biotech plants. Of course, we have increased from the start year when they start to commercialize the genetic modified plant. We can see that there is an increase in the cultivating area, increase about 2.7 billion hectare from the 1996. The second and the most applicable technique right now is the genome editing. So what is the genome editing? Genome editing is a very precise technique to modify the sequence of the huge genome. We can choose a specific nucleotide in the genome to modify this sequence and to regulate the gene expression pattern. So we have to predetermine first what we want to do. So if we want to do work in this gene, we'll have to have the sequence of this gene and specify which part exactly we want to modify. So the genome editing can be used to insert, remove, or delete or replace different parts in the genome. It depends on the nuclease. Nuclease is used to break the nucleic acid. So we have different kind of nuclease that cause damage in the DNA, and we depend on the repair system of the cells to repair this damage. There was several trials to use genome editing, like zinc finger, like TALEN, like meganuclease, but the most promising and most common use right now is the CRISPR genome editing. So the other 3 kind depend on protein to identify the sequence, but in the CRISPR, they depend on RNA to identify the exact sequence that we need to modify. So for the CRISPRs, we have the enzyme itself and the guide RNA, which identify the sequence in the genome. The enzyme will break the DNA in double break in the specific site. And we depend on the cell itself to recover the damage. Because if the damage will not be recovered, the cell will die and we will lose the mechanism. So if we have a template, similar to the part which is going to be breaked, we'll have like a switch between the damaged part and the desired DNA that we want to replace. If not, the cells itself will do like deletion or insertion and make the ligation again to have the chromosome, the entire chromosome repaired. This we call it non-homologous end joining, and this we call it homologous direct repair. Of course, both of them, this will lose the function of the gene. We can here replace part of the genome or part of the gene with the desired sequence we want. If we want to make a big replacement, we can do double break, but in different strands, in a part sequence. We use mutated nuclease. This one will break the first strand, the other will break the second strand. And we will have, of course, a template which can be replaced, crossing over and switch the part that we want to introduce. So genome editing can be used to modify the gene. Not only for that, we can also use it for activating the gene, like you fusion the nuclease with the activator, or suppress the gene, or locate the desired sequence inside the genome. So in this three part or the three example, the enzyme will lose its function as a nuclease. It's only used for the binding and for forming the character that we want. So for delivering of the target DNA for the enzyme and for the RNA, we have to insert the DNA inside the cell. So we can use protoplast, can use agrobacterium-mediated transformation, we can use DNA gun. Those three techniques depend on tissue culture. So we need tissue culture for genome editing if we use one of this technique. But if we use virus-mediated delivery, so we will enter the sequence inside the virus genome, we infect the plant with the virus, and in the second generation, we will not have transgenic plants. So in this case, we can use agroinfiltration with agrobacterium or from protoplast. We insert the DNA, and the DNA will form its editing, and we will have the edited plant. For the virus, like TRV viruses is an RNA, we can introduce the target guide RNA and agroinfiltrate or infect the plant with the desired constructs, and we will choose or select later the edited plant, which is not transgenic plant, but it will have the mutation or replacement that we want. The other kind is we can have transgenic plants, but the transgenic plant will enter the DNA in one chromosome, and we hope that the gene that's going to edit is another in a part sequence. So in the second generation, we can get rid of the transgenic insert we introduce, and we'll have the edited plant. So it also needs tissue culture, but by the end, we will not have a transgenic plant. So genome editing is more efficient and precise. It could generate new varieties without introducing foreign DNA. By the end, we will not have any foreign DNA. That's why it's considered non-transgenic crops. So it can have different modification like insertion, deletion, point mutation, replacement, and so on. And we expect that it will not have a restricted regulation like the transgenic. So is there a edited plant in the field? Yes, we have. For example, in 2012, we have edited plant, but it's by TALEN-based editing for disease resistance. We also have potato plant for less acrylamide, so it's less browning, and it's already produced in 2015. We have also a disease resistance, like sensitive gene. Usually when we want to develop disease-resistant plant, we use the R gene, increase the expression of the R gene. But in this technique, we suppress the expression of sensitive gene which allow the microbe to enter inside the plant and infect the plant. For instance, for blight, late blight resistance, we can knock out the S gene and we'll have resistance against the late blight. Sorry. Also, we have they have developed modified plant against Xanthinomas, and it's already in the market. It's already tested and it's ready for marketing. We have genome editing of wheat, altering the spike and grain characteristic, and this was in 2020. It's a very new one. And also this is a nice one. I like it because it has different color of Petunia, pink, purplish-pink Petunia. They change the color. This is for commercial purpose, for cut flour production. Also, we have drought-tolerant tomato. It's already there by mutating a gene for specific that allow the plant to tolerate drought in tomato. It's not the stomata gene which allow drought tolerance. It's another kind of gene. We have genome editing for canola for increase the yield. This was published in 2021. It's already there. And also we have the high yield enhanced aroma in rice by by editing 3 genes. So editing is not necessary to do gene by gene. We can do different genes by introducing large different part of guide RNA that will allow editing of different genes at the same time. This is very important because some of our children, they have a problem with wheat because of the gluten. So gluten-free wheat is very expensive and very hard to develop. So they used genome editing in 2019 to remove the gene which produce gluten, and we have safe plant for people that have allergy against gluten. Also for fire blight, they have been used in apple for resistance. So there is different advantage and disadvantage of tissue culture. The advantage of tissue culture is the availability to raw materials. So some of the plants are difficult to cultivate. So we can use tissue culture to propagate them in a mass production, and you can use it in different fields. Like for instance, in Egypt, we have one plant, we call it ras. It's a tree plant, medicinal plant. We have only one plant in Sinai. So we use tissue culture to micropropagate this plant and recultivate it again in the same environment, in the same place. So we can use it for developing drugs. For fluctuation in supplies and quality, like production of crude drugs and so on. For new methods for isolation of new component from plants, we can use it for introducing different components like vaccine and different materials and develop plant with this product. For genetically modified or editing plant, we can use it also for use the tissue culture. For producing disease-free and so on, and like biosynthetic pathway for plant preservation. We can preserve the plant if we, for years, if we have different cultivars that right now is no longer are used, we can preserve them. Maybe later we're going to use it in the commercial purpose. For biomass production, of course, this is a very big industrial process are used. For medicinal compounds can be used also. For natural compound can be produced. For medicinal plant, we can use medicinal plant, improve them, and have a lot of product from the medicinal plant, and we can propagate it without seeds. So what is the disadvantage of tissue culture? They have high level of expensive is required, like equipment and so on. If any error happen during the tissue culture, like somatic clonal variation, that will collapse all the plant will produce. So if we have one mutation from the start of this plant, we'll have all the propagated plant have the same mutation. So we have to avoid any somaclonal variation or any mutation or any undesired characteristic during our tissue culture propagation. It needs a lot of chemicals for preparation for an artificial media, and we need to adapt a protocol for individual plants. So in the beginning, it will take a lot of time to make the suitable adaptation. And then once we have adapted this technique, we can use it for multiple propagation. the instability, because if we cultivate it in a different tissue culture for several times, we will lose the activity of this plant. Of course, it's slow growth, take a long time to have tissue culture, expensive process as well. So by that, I would like to thank you for your time and for your attention for the lecture, and I'm open for any kind of question if you want. Thank you.
Thank you, Dr. Najla, Dr. Samar, and other participants. If any of you have any questions, kindly pitch in.
Khazima, we were receiving questions continuously on the chat, but I invite all the participants, if you can ask directly, if you want to ask anything directly to Professor Nagla, so she's here to answer your questions. Okay, if you have no questions, then thank you so much, Professor Nagla, for very valuable and informative lecture. And Khazima, should we proceed to the second part?
Yes, sure. With this, I would like to thank Dr. Najla for your time, and it was a very comprehensive presentation. And with this, we move on to our hands-on training part for day number 2. And this practical session is on tissue culture of cash crops, which is rice. With this, over to you, Dr. Samar.
Okay, thank you so much, Khazima. Yesterday, we focused on the tissue culture of ornamental plants, and we used an example of orchids. Today, we will try to give you overview of the tissue culture of rice, that how you can use rice seeds to tissue culture rice plant. So based on that, we will start with some of the basic preparation which we presented yesterday in order to
with 2 filter paper, tools and apparatus for tissue culture experiment. Apparatus required for tissue culture are forceps, scalpel, and blades, Petri plates with 2 filter paper. All the apparatus should be in spotlessly clean condition. Wrap scalpel, forceps, and blades in paper, brown paper, or newspaper. Wrap battery plates Put cotton in the jars, which will be used to clean the laminar flow hood. Fill the bottles with deionized water, which will be used to sterilize explant. These things are now ready to autoclave and will be used for tissue culture experiment later on. Media preparation. Preparation of stock solutions for MS media. Weigh desired amount of macronutrients in 1 liter of distilled water, one by one, and store in refrigerator for further use. Weigh desired amount of micronutrients in 1 liter of distilled water, one by one, dissolve them properly, and store in refrigerator for further use. Prepare all the stock solution and store them for further use for the preparation of MS media. Preparation of MS media. Take distilled water from distillation unit. Distillation unit comprises of 2 units, deionized water and distilled water. Take 700 ml of distilled water for preparation of 1 liter of MS media. Turn on magnetic stirrer and put 700 ml of distilled water in a beaker on the stirrer, and also add a magnetic bead into the stirrer to dissolve the nutrients properly. All the stock solutions of MS media are now ready. First, add 50 ml of macronutrient stock solution into the MS media. Add 1 ml of micronutrient stock solution with the help of micropipetter. Add 1 ml of vitamin stock solution in 1 ml MS media. Add 5 ml of EDTA stock solution. Weigh 30 grams of sugar or sucrose for 1 liter of the media. Now add 30 grams of sugar into the media. Adjust pH of the media to 5.7 or 5.8 with 1 molar NaOH or 1 normal HCl. Make up the volume of the media in a measured cylinder. Make up the volume up to 1 liter with distilled water. Weigh 8 grams of agar, which we'll use to solidify the MS media. Add agar in MS media solution. Microwave the media to dissolve the agar completely for 10 minutes. Add phytohormones like auxin and cytokinins according to the plant requirements. Media is now ready to dispense in 250 ml glass bottles. Each jar contains measured quantity of 30 ml of MS media. Close the caps of the jars carefully and tight. And proceed to the autoclave. Turn on the autoclave and open the lid. Place media jars carefully in the autoclave. Close the lid properly and check the outlet valves. Set autoclave program for 30 minutes at a temperature of 121 degrees Celsius and pressure of 15 psi. After autoclave, the media jars will be kept in growth room for 3 to 4 days to check any sign of contamination. In case you find any sign of contamination in your media jar, remove and discard that jar immediately. Contamination-free jars will be used for further experiment. Media jars can be stored at a temperature of 20 to 22 degrees Celsius. Process of removal of the contaminated jars from the growth room. First of all, collect all the contaminated jars and autoclave them. After autoclave, media should be collected in a plastic bag and proceed to discard. The jars are then washed and dried. Cleaning and preparation of laminar flow hood. Wash your hands properly with hand wash or soap before entering into the tissue culture lab. Before starting your experiment, you need to put on your personal protective equipment like lab coat, mask, pair of gloves, Laminar flow hood is used to maintain the sterility of the cultures, and in order to maintain the sterility, we need to sterilize our hood first by exposing it with UV light for 15 minutes. Turn on UV light and cover the laminar flow hood, as UV light is hazardous for health. Turn off UV light, remove the cover, turn on blower and light. Spray your hands with 70% ethanol before starting your experiment to disinfect them properly. Disinfect the surface of hood with 70% ethanol. Spray the surface of hood with 70% ethanol and wipe all the excess ethanol with autoclaved cotton. Disinfect all the objects with 70% ethanol you are planning to use during your experiment or need to bring into the hood, like media jars, autoclaved battery plates, sterilized tools. Put the tools into the sterilizer and turn on the sterilizer. Laminar flow hood is now ready to use for tissue culture experiment. In order to turn off the hood, remove all the waste and used Petri plates and media jars. Turn off sterilizer. Disinfect the surface of hood with 70% ethanol. Remove all the excess ethanol with autoclaved cotton. Turn off switch, light and blower and cover the laminar flow hood. On day 1, we performed tissue culture of horticulture crops initiated from leaves. Today, we will perform tissue culture of cash crops initiated from seeds. Selection of explant. Explant can be leaf, stem, embryo, hypogotyle, cotyledon, or meristem of a plant. Selection of explant and surface sterilization are most important to make explant free from contamination. To initiate tissue culture, select fresh and healthy seeds without any visual disease symptoms. Initiation. Turn on light and blower Before starting your experiment, wear a pair of gloves and sanitize them with 70% ethanol before starting your experiment. Clean the surface of hood with 70% ethanol and wipe all the excess ethanol with autoclaved cotton. Sterilization of egg plant. Egg-plant sterilization is the process of making egg-plant contamination-free before establishment of cultures. It is important that egg-plants be free of any contaminants including fungi and bacteria prior to tissue culture without losing their biological activity. Again, sanitize your gloves with 70% ethanol before starting your experiment to avoid any kind of contamination. Add ethanol in the selected seeds and dip them for at least 30 seconds in ethanol. Shake a little so that seeds get fully exposed to the ethanol. After 30 seconds, discard ethanol carefully in a discarder. Now add 20% commercial bleach, that is sodium hypochlorite, for 10 to 15 minutes. With constant shaking so that seeds get properly exposed and sterilized. After 15 minutes, drain all the liquid in a discarder carefully. And wash the seeds with autoclaved distilled water 3 to 4 times to remove all the traces of bleach and ethanol. After sterilization of egg plant, clean the surface of hood with 70% ethanol. Spread the seeds in a Petri plate to soak all the liquid. Inoculate sterilized seeds into the MS media one by one with the help of forceps. Avoid crowding in the jar and do not use hot forceps as it can damage your egg plant. Seal the jars with parafilm tightly to avoid any kind of contamination. Label the jars with the type of egg plant, type of media, and the date. These jars are now ready to be transferred into the growth room. These jars are now kept in growth room with illuminated condition of 16 hours of light and 8 hours of dark at a temperature of 24 degrees plus minus 1 or 2 and relative humidity of 40%. Sterilize the surface of the chamber before putting your culture jars with 70% ethanol and wipe all the excess ethanol with autoclaved cotton. After shifting your jars into growth room, keep visiting your growth room after 2 to 3 days to check any sign of contamination in your culture jars. In case you find any contamination, you have to remove that jar immediately from your growth room. And keep the contaminated free jars in the growth room for further growth. Subculturing is the second stage of tissue culture. After 2 weeks, when the cultures develop, increased shoots are achieved by subdividing the cultures. The media at this stage has more cytokinin present than during initiation stage. When optimal number of shoots are developed, rooting is derived by lowering the cytokinin level and increasing the oxygen level in the media. These plants are now ready to be subcultured in fresh media with specific hormones. Take your tools out of the sterilizer. Open the jar and carefully take out your plant out of the jar. Do not use hot forceps as it can damage your plant. Remove dried dead parts before shifting them into the fresh media to avoid any kind of contamination, and make one sharp cut to minimize the release of phenolics. Once the dried dead parts have been removed, inoculate the plants into the fresh media with cytokinins. Avoid touching the walls of the jars to avoid contamination, and do not use hot forceps as it can damage your plant. After covering the jars with the caps, seal the jars with the paraffin to avoid contamination or to minimize the rate of contamination. Label the jars with type of plant, type of media, and date properly before shifting them into the growth room. These jars are now ready to shift into the growth room. Growth room conditions. All cultures are maintained in a culture room at a temperature of 24 degrees plus minus 1 or 2 with 16 hours photoperiod, and subcultures were performed every 21 to 28 days to provide a new fresh nutrient under the same condition. Before placing your culture in the growth room, sterilize the chamber with 70% ethanol and wipe all the excess ethanol with autoclaved cotton. Transfer the jars in growth room under illuminated condition of 16 hours of light and 8 hours of dark at a temperature of 24 degrees plus minus 1 or 2 with relative humidity of 40%, and keep visiting your growth room to check any sign or any kind of contamination in your culture jars. Acclimatization. It is the next step after subculturing. The term acclimatization is defined as the climatic adaptation of an organism, especially a plant, that has been moved to a new environment. This procedure consists of removing plants from the test tubes or culture jars and then placing them in growing media, that is peat perlite and topsoil in ratio of 1:1, watering and fertilization well, and placing them into an ambient greenhouse environment or an acclimatization chamber. After tissue culturing, the last stage of micropropagation is called transplantation or hardening in the greenhouse. This step is called acclimatization because before exposure to the direct sunlight in the field, plants should be grown in the greenhouse. Take out the plants from the media jar. Wash the traces of agar from the media with tap water. After rinsing it with tap water, they should be dipped in fungicide to avoid any kind of fungal infection before transferring them into the field. Again, rinse the plant with the tap water. After proper washing, these plants are now ready to be transferred in the greenhouse for acclimatization purpose. These are the acclimatized plant in the greenhouse.
So this was the end of the technical videos of our today's technical session. Now I would like to request Khazima to read the chat box and some questions from them, and our technical team will answer that.
Thank you, Dr. Samar. Dear participants, I'm going to read out some questions and we'll take your name. In case you want to, ask them in detail, you are welcome to participate. So let me ask the most recent question. Okay. So we had a question from Ms. Aksa Amin. This is basically a continuation of our previous question, but if gases produced in result of photosynthesis are not released when jars are sealed, plant can die. So Dr. Fahida would probably be the right person to answer her.
Fahida, yahan aa jao.
Yes, Aksa, I already informed you in the chat box that plants that are grown inside the media, they already have the nutrients that are helping the plants to do the photosynthesis inside the jar. And also there is the proper temperature is controlled and the humidity, and with the proper photo period, that is a 16 hours of light and 8 hours of dark in the growth room, that are helping for plants to do the oxygen and take the, that's why I think that these plants can be grown easily and there are no release of gases inside the jar because they can recycle the gases inside the jar. And the media provided inside the jar, they help the plants.
Okay, thank you, Fahida. So let me ask one more question. This is from Salman Kashmiri. Do we have to keep vacuum in the UV chamber? So any one of you from the ICCBS team can answer him.
Vacuum in the, I think we have the laminar flow hood. For once we expose the plant to the, expose the material to the UV, But vacuum is used for different purpose. For tissue culture, we don't use the vacuum. That is used for drying of the certain kind of explant.
Okay, thank you. We have another question, and this was again from Ms. Aksa Amin. How to choose a plant for tissue culture research? So this is a general question.
Sorry, could you repeat, Khazima?
Yeah, we have a question from Ms. Aksa Amin. She's saying how to choose a plant for a tissue culture research. So what I'm getting from our question is that what are the model plants that we can use for tissue culture research, or what are the traits that we are looking for a plant which we can use for tissue culture research?
Yeah, actually, in first today's lecture, the teacher, they also mentioned clearly that the ability to grow from any part of the plant. That is the reason that plant is totipotent in nature, and it can be grown from the stem, root, or any kind of embryo and seed. So that is the call as the direct organogenesis. Hence, the plant can be grown from any part of the plant, but it depends on your nature of the experiment and your objective, that what you want to do further for your experiment.
Aksa, let me elaborate the question. In my understanding is that you are asking that which type of plants can be used for the tissue culture. So let me tell you that science is always for a purpose. So that purpose need to be decided by you, that either you wanted to do the tissue culture of an ornamental plant, or you wanted to do a tissue culture of a medicinal plant that will be helpful for those who are using that plant for to cure a disease or something like that, or you wanted to do a tissue culture of any crop in order to facilitate the humanity. So you can do the tissue culture of any plant, but choice is yours and the aim would be decided by you.
Thank you, Dr. Samar, and your team member who answered Ms. Aksa. So there is another question which I would like you to answer as well. Is there any need to develop tissue culture technique as Pakistan is already an agriculture country? So this is from Arshia Musharraf.
Okay, Pakistan is an agriculture-based country, but problem is that with the passage of time, we are suffering with the problem of volume of water. Our lands are going to be suffered because of this same and thaw, and many other problems like attack of pest and other diseases on these crops. And because of these, the production of each crop, including from cotton to wheat and rice and all others pulses, they are going to be reduced with the passage of time. So the tissue culture is the need of time, which need to be adopted and to be accelerated in order to facilitate our farmers, because they are suffering with the poverty, and in addition to that, in order to support the economy of our country.
Thank you, Dr. Samar, for a very comprehensive answer. So there is another question from Mr. Ahmad Shahab. He's asking which is better choice as explant for a tissue culture, either developed from a seed or from already established tree plant.
Yes, for the explant selection, I also already mentioned before that it depends on your desired experiment, whether you want to grow it for micropropagation or you want to grow for any experiment. For example, in the case of transformation, you need to grow the callus first. That is called as indirect organogenesis. So for that purpose, you need to choose the leaf or stem part for the explant, and then you can proceed it further for the callus induction. And if you want to do the micropropagation, that is used for the commercial purpose, and to sell it for the marketing purpose, then you can do easily from the seed. Then why we need to go for the hard method? Because from the seed is much easier to grow in the tissue culture than the other explants.
So thank you very much again. With this, let me ask another question. This is from our participant, Tolshi, and he or she is asking, is it okay to autoclave MS medium after adding hormones? So any one of you can answer this.
No, I think yes, because hormones are heat sensitive and they need to be filter sterilized. So they are already sterilized and they don't need to be autoclaved, because autoclave temperature degrade the hormonal activity, and in the end the plant growth will be affected. So hormones should be added after autoclaving. It is recommended method.
Thank you very much. So I think there is another question in continuation to what has been said about using tissue culture in an agriculture country. So she's asking, would it be cost effective to have plants from or agriculture from tissue culturing techniques?
Is it cost effective?
You may say it is cost effective, because in the field, if you are growing the plant, it takes much labor and time as well. And also you need more amount of water and other kind of fertilizers and pesticides, because the outside environment can affect the growth of the plant. But in the tissue culture, it will be much feasible to do once you have established. I mean, for the establishment, obviously it takes a little cost, but once the tissue culture setup is established, then it is much cheaper to mass propagate the plants, and that would be the disease-free plants. क्या करना पड़ेगा? Step-by-step requirement for tissue culture. Can you please see already? लेकिन feedback का तो और further processing. Can you use PCR oil?
So thank you again. We have another question from Aruj Arshad. While producing BT crops, we produce better and pest-resistant crops, but seeds are sterile. Has anything done to help this disadvantage?
Khazima, could you repeat the question, please?
So this is a question from Arshia, and give me a minute, please. So Aruj Arshad, and she's asking, while producing BT crops, we produce better and pest-resistant crops, but the seeds are sterile. Has anything been done to help this disadvantage?
No, I don't think so, because like in the case of hybrid seeds, because most of the crops now are diverting towards the hybrid technology. So these seeds are also sterile, but the main purpose for producing the modified crops is that we need to get the more production. In the end, we are interested to get the more product. So I don't think that the seeds are important to get as a sterile. Sorry. हाँ सबके लिए एक ही.
Thank you very much. I think we can take one more question, and this is a question from our participant from Malaysia, I think, Noor. What is your opinion on GMO food? Some opinion says that it is bad for health or for the ecosystem balance. What do you think?
Yeah, I think so, but now as you can see from most of the publications and inventions from different scientists in all over the world that most of the crops are now produced through the genome editing, because in the genome editing, there is no foreign gene involved. So the gene is edited inside the same organism. Hence, it is not toxic for the plant. Yes, in the case of GMOs, I can say that the gene can be expressed later and it can be toxic to the health in the human body. That's why in Pakistan, we have only approved BT cotton, because BT cotton is not consumable inside the body. That's why it is a GMO, but can be used for the enhanced production of cotton.
Thank you again. With this, I would ask Dr. Samar if you can share
Yes,
some
Khazima, we have
videos.
a very short 2 to 3 minutes video of the tissue culture facility of the ICCBS. And after then that, we will proceed for the conclusion. This is the video of our greenhouse and hydroponic facility. Yes, Khazima, over to you.
Thank you, Dr. Samar, for this very fruitful course. And with this, we come to end of our technical session for today. I would request Professor Dr. Iqbal Chaudhry, Coordinator General Comstech and Director ICCBS, to say a few words about this 2-day training course and its usefulness to its participants. Over to you, sir.
Bismillah ar-Rahman ar-Rahim, Khazima and Dr. Samar, and all the participants, Assalamu alaikum wa rahmatullahi wa barakatuh. You see, I can gauge the interest and enthusiasm of all the participants in this very important skill development course. This has been one of the most popular events of comstech in collaboration with Pakistan Biotechnology Information Center and International Center for Chemical and Biological Sciences. And I can see the participation from 3 different continents. So it's widely attended, and hopefully widely benefited event, which I believe would lay the foundation of much more events in the future also. I consider this as one of many events which my colleague would continue to organize in this very exciting field. Plant biotechnology, as we understand, is a field which is directly relevant to human welfare. And as we see sustainable development goal, a lot is dependent, whether it's health or empowerment or global poverty, are related to the availability of food and a proficient agriculture sector. Climate change has placed agriculture at a very disadvantaged position because the new realities of climate change is forcing people to go for with different kinds of avenues, and technology is certainly a very important avenue for them to follow, because technology offers solution of many of the problems which humanity face today, not only because of pandemic, but also because of climate change and long-term uh, climate variations which require resilience and also. So with this, I'd like to thank all of you for your very active participation. I'm sure that you have benefited from this event, and I am confident that you would continue to interact with each other and with my colleagues in Comstech and also in ICCPS, and provide your feedback. Tell us how these events can become even more useful and need-based, and this would actually help us to design more courses. And as mentioned by speakers yesterday, we all are looking forward to a day when we'll all be able to meet in person, and this journey of acquiring knowledge and this joy of learning would continue. Thank you very much. Thank you.
Thank you, sir, for your kind words. With this, we come to end of our two-day course on plant tissue culture. At the end, I would like to thank Dr. Samar and her team at ICCBS for just organizing such a wonderful online course. And of course, I'm really, really grateful to our participants for their continuous support throughout these 2 days. And with this, we are hopeful to see you in our next program or in next event, be it a webinar or an online course. Thank you very much again for joining